GCN5, ESA1, and CHD1 more than this transcriptional regulators?
Thanks, Sean, and thanks, everyone. So the work I'm going to be talking about was all done by former graduate student Lynn Andreas in my lab. And what we're interested in is DNA repair, but really the recognition and step of that. And this requires opening the chromatin in more heterochromatinized regions. And so we're interested in trying to find those factors that are involved in opening up for repair, but not involved in transcription. And like you just heard, Tetrahymena is a good model for studying this because of the two nuclei. So I don't need to go into too much of this because Alejandro just presented some of this, but we're really interested in that kind of regulation of DNA accessibility for repair. And it's known that there are histone modifications that do this process, and for us we're looking at histone acetyltransferases. And the two I'm going to talk about are GCN5 and ESA1, and both those add acetyl groups. And they're involved in also then promoting the recruitment of ATP-dependent chromatin remodeling complexes, like the SWI-SNF complexes that we just heard about. So just a little background on these two acetyltransferases. GCN5 is found in Saga, where you see ESA1 in yeast is in NUA4, and both of these are histone acetyltransferases but have different functions. Now GCN5 is kind of there for that activation during and allowing transcription like we just heard. And what I'm interested in is it's been shown to potentially allow the admittance of the repair mechanisms to damage but not allow transcription at those spots. And to study that more, that's kind of our goal. And then after DNA damage, acetylation of histones by GCN5 is decreased and then increased back up. So we're going to be looking for a gene that kind of does this pattern of maybe decreased and increased acetylation. Now, where ESA1 is that it's in that NUA4, it's increased locally in cases where GCN5 acetylation is decreased. So they're usually opposite. And there are proteins in Tetrahymena, like you just heard about, that are involved in these. And now, when we did our initial bioinformatics, we found 3 orthologs in Tetrahymena of ESA1. I've listed them here. When we do a lot of bioinformatics, the best we can kind of pull out from bioinformatics is the third one that I've started. That's what we're going to call ESA1. Until we see functionality, we can't really officially say that. But and some of the other ones look like TIP60 or MST1. Now, the other protein that is in my talk, and again, we'll see how much time we have to go through all these, but the chromatin remodeler that we're kind of interested in is this CHD1. This is a SNP2 related. It has chromatin binding activity as well as a bromodomain, and it can be recruited to sites of acetylated potentially during DNA damage is where we're looking at. And it's involved in that histone exchange and assembly. And when we were looking for transcription coupled repair proteins, and the closest thing that came up to a RAD26 is this CHD1 too. So, and I'm going to explain a little bit of that during my talk. I don't have to go into this too much, but why we use Tetrahymena, I used to do stuff in yeast in DNA repair, but that nuclear dimorphism really plays well to, especially if you're studying silent DNA repair. So throughout my talk, I'm going to talk about damaging these cells and looking at different repair processes with 3 different damaging agents. We're going to look at UV for nucleotide excision repair, MMS for looking at translesion synthesis or double-strand break repair, depending on the concentration and the amount of time you can get, sometimes more basic excision repair or double-stranded breaks. And then we're going to look at hydrogen peroxide, which will give a more generalized release of oxygen radicals and be involved in basic excision repair. So first off, whenever in our lab we look at trying to find the homologs, we do some phylogeny. And when you compare the Tetrahymena GCN5 to other organisms, GCN5s, this is what it looks like. It looks fairly good. And we always root it to something, and we're using ESA1 Tetrahymena here to show that they are distinctive. And you can see that the paramecium and the Tetrahymena are kind of grouped with some of the fungi and plants, and in a bigger grouping with vertebrates. Now ESA1, it's more distantly related, and you can see that it kind of just groups with all of the major ones from other organisms, but pretty far away, and GCN5 is way out there for rooting. Now when we look at the CHD1 homologs, and I'll show a alignment with the domains in just a minute, but Tetrahymena and paramecium come off similar to the GCN5 with some of the higher eukaryotic ones versus fairly farther away from Saccharomyces. And again, I put Rad26 on there because we are interested in double-stranded, nucleotide excision repair, both transcription coupled in. And there is no good Rad26 homolog in Tetrahymena. It might be because the macronucleus isn't necessary to have transcription coupled repair when you have a number of loci, or something else is covering that function. So here's our diagram of the domains, and if you look up here, you can see that you've got the chromodomains, the ATPase, and then a bromodomain in the Tetrahymena. CHD1, it's a fairly large size compared to, say, the yeast. And you can see, when you look down here, this is Rad26 that doesn't have some of those things at the N-terminus. And it goes well with some of these other ones. So it's a fairly good hit for a homolog with CHD1. Now I'm going to talk now about our analysis of trying to find the regulators of repair chromatin and opening like chromatin. And so first off, I just wanted to show a little bit of data of GCN5 expression. If you look at the microarray and the microarray data, GCN5 does peak at about 3 hours. We're seeing in conjugation, and then in starvation you see it going up. What we did when we looked at it, we first did a short time course, saw it increasing. Then we did a longer time course, and you can see that it does kind of go up and then come back down, and with its maximum after starvation around 6 hours. Now, because we're looking at trying to find things involved in DNA repair, we looked at after damage to UV, MMS, and hydrogen peroxide. Now, if you look at the scale, these are not very large scales, but when we've done this at least 6 times, we can get this kind of consistent down regulation after UV, and then there's this kind of weird pattern of up-down for MMS. And you're going to see a similar pattern when we see a couple of the other genes. And then with hydrogen peroxide, we see this down regulation. And this seems to happen sometimes with GCN5. There's a turning off transcriptionally to then localize it specifically to certain places after repair from other organisms. So I wasn't too concerned that it doesn't have as drastic. So we're going to have to go to the protein to kind of get more information there. Now, if we look at the ESA1, again, this down regulates after DNA damage, although it goes up then after MMS, so more double-stranded break maybe process. And then it really doesn't significantly change at all during hydrogen peroxide. So this may have a role in double-stranded break repair. And then CHD1, and we're looking at, when we look at these, we're looking at patterns of having similar patterns of expression to each other. And so if we look at the GCN5, and we look at the CHD1, we see a similar pattern where we see it go down and then come back up after UV. With MMS, it goes down a little bit, but then not significantly in the later times. And then with hydrogen peroxide, though, this one, it actually increases, but it's fairly variable, and so maybe not significant. So after this, that didn't tell us too much, so we needed to make some knockouts and then some overexpressing strains. And for GCN5, we start with GCN5, and anytime you see the red, that means overexpression. The blues are knocking downs with an shRNA construct, so that we can keep oriented. And when you look at this, you can see there's these cells that overexpress, and these are only under the MTT promoter. So you just turn it on for an hour, and then they pretty much are very sensitive to hydrogen peroxide, and they are slightly sensitive at the beginning to little lower doses of MMS, but higher doses, they seem to be more resistant. And so that's what I was talking about. There's a difference between the amount of MMS causing basic excision repair versus double-strand break repair. And if you look at when you knock it down, there's actually, again, a fairly dramatic decrease in survivability after hydrogen peroxide. And in MMS, there's a resistance to MMS. And so this is not uncommon, because sometimes one pathway gets heightened when another pathway is inhibited. And so sometimes we see this with NER and double-strand breaks and things like that. So it looks like it might have a role in protecting from oxidative radicals. And so some of the other things we looked at, we wanted to look at growth rate of these cells. And you can see that when you knock down GCN5, they have a very severe defect in growth. And if anything, when you overexpress it, they grow a little better. So. And when we want to look at kind of survival after starvation, because what we did is we put these in stock tubes and then they died. So, and that's how she did the transformation many times, and I thought she wasn't keeping her stock tubes up. But they wouldn't even last a month in a stock tube, let alone a couple weeks. And what we found out is that her overexpressions, even when they weren't being induced, and this was uninduced and then induced, they're still kind of having a phenotype of less survival rate in starvation. So. And even the knockdown isn't got much of a phenotype, though. Now, because of this oxidative radical kind of issue, we wanted to look at SOD2, which is involved in the repair pathway of oxygen radicals, and we did hydrogen peroxide as a control, just going to show an upregulation of SOD2. But if you look after starvation, you have a nice increase of SOD2, kind of to protect from oxidative radicals. But in both and in the red here is the overexpression ones, you have it higher at the beginning, and this is all normalized to the growing levels for that time. So it actually doesn't, it just is up already at 6 hours, but it goes down over 48 hours. And the same thing happens with the knockdown. So both of these have phenotypes of being sensitive, and it probably has to do with if you have too much or too little, it's causing it to not regulate these genes. And we did look at the oxidative radical accumulation, and I'm just going to tell you, because I don't have time to talk about it, that it did increase inside the cell. So we did an assay to look at ROS inside the cell, and it did increase. Now, just to finish up to look at the knockdown of ESA1, these guys have a increased protective effect of MMS, so they're more resistant to MMS, and actually more sensitive to UV, which makes it look like maybe this one is the one involved with nucleotide excision repair. So that's the one that we're going to be pushing towards, looking at for nucleotide excision repair and global genome repair. And then for the CHD1, This follows a lot like the GCN5, and if you look at MMS treatment, though, it's very sensitive to MMS, and for UV, it's more resistant. So this is like a protective effect when you don't have it around as much in UV. And for those CHD1 knockdown cells, we noticed that they were growing really, really slow. And if you look at it, they have an increase in doubling time, and their diameter of their cells are a lot larger. When we look at it put it through a culture counter, we can get the size distribution. And here's the distribution is really pretty drastic in the size change for these knockdown cells. And this is just a summary that GCN5 overexpression and knockout severely make them sensitive to hydrogen peroxide. Overexpression and knockdown make them resistant to higher doses of MMS, and the knockdown cells have slower growth. Overexpression causes decrease in survival during starvation, and then overexpression and knockout have higher levels of SOD2, and like I said, and it inversely correlates with ROS, which is data I couldn't show you. And ESA1, the knockdowns are resistant to MMS at higher doses, but sensitive to UV. So these look like they might be involved in repair at nucleotide excision. And then lastly, CHD1 knockdowns are sensitive to MMS and UV at low doses, but then, and I didn't get time to talk much about, but but resistant at higher doses of UV. So. And just one of the students in my lab made a cartoon. So our lab studies chromatin and DNA repair, and that's our mascot. So, any questions?
This might be a naive question, but how did you show that activity and expression are equivalent?
Activity and expression. Well, we haven't done any look at activity of the enzymes yet. We are in the process. We have tagged versions, and I didn't get to show that either. We have localization stuff. All that is on slides after in case you want to see that later. But yeah, we have tagged them with a flag hiss, and we're in the process of trying to purify them and look at now in these knockdowns and overexpressions the modifications and see after repair what are the changes in these modifications. So a lot of people are looking during transcription, but we want after repair.